I'd now like to invite on stage our next speaker, Dr. Vineeta Bal from Indian Institute of Science, Education and Research, Pune. I request our moderator to introduce the speaker. Thank you.
Okay, so they've handed me something so I don't totally mess up. But anyway, I won't look at it, so you have some entertainment. So again, it's a pleasure to introduce Dr. Vineeta Bal. So she did her MBBS in the University of Pune, and then did her MD in the Hafkind Institute, and went on to the MRC unit which studies tuberculosis in London for postdoctoral work. And then after that, she came back to India to join the National Institute of Immunology, where she has spent really the bulk of her career working on T-cell immunity and the role of T-cells in controlling infection and their development and so on and so forth. And she is currently an emeritus professor at ISER Pune. Okay, so please welcome Dr. Vineeta. Yeah.
Thank you. Thank you to the organizers for inviting me. Arvind's talk and my talk are going to be completely different. Arvind actually talked not so much of darkness. Arvind talked primarily based on his work, reviewing many years of work. And the topic I was given was diversity in the immune system. We have worked on it this bit about 20 years ago, and there isn't a career which has gone into it. So my talk is actually going to be really basic teaching. I also wondered why organizers wanted to have somebody talk about generation in immune diversity, but since I was told if I could come and talk, I am going to do it. So the diversity is what we are going to talk about. Essentially, what is immune diversity? It can be thought of in many different ways, and that's why I'm putting this forward as information, that its ability to use diverse immune mechanisms for mounting immune response. So it is obviously something that we do need. Complement is one way of immune response, complement activation. There is antibody response, T-cell response. So these are very diverse responses that are very much required for successful defense of the body. There is also a great diversity in the immune cells. So while complement is a soluble component of the innate immune system, there are macrophages, neutrophils, B cells, T cells, NK cells. So we need, again, all this diversity in the immune system to mount a good immune response. And there is also the diversity which is thrown at us in the form of pathogens. Pathogens are innumerable. We also encounter new pathogens like SARS-CoV-2. So that kind of diversity is something that we should be able to deal with. That's the diversity. Is that the immune diversity? In a sense, yes. So in order to achieve all of these, and even maybe more subtle nuances of this, and generate innate and adaptive immune cells, recognizing potential every pathogen in the world. So every pathogen in the world is the important point that I wanted to make, and I will take you towards that. But essentially, I'll show you a little bit about differences in innate immune cell diversity and adaptive immune cell diversity. That will remain the focus. So primarily now, beginning with innate immune cell diversity and recognition, the way they do it. So these cells are neutrophils, monocyte, macrophages, dendritic cells, and so on. So they are the ones which are very much rapid response cells which reach the site of invasion by the pathogen. And when I say invasion, we have all sorts of barriers to the body like skin, the mucosa. As soon as the barrier is breached, I'm calling it as an invasion. And with invasion, these cells do come into sort of start moving if they are not already there. And there is a need to recognize pathogens of diverse nature. One doesn't know at what time which pathogen will enter from which location in the body. We have a huge body. So essentially, that's the diversity which we have to keep in mind. And the receptors on the innate immune system recognize shared elements of groups of pathogen. For example, cell surface mannose, lipopolysaccharides, single-stranded RNA, double-stranded RNA, and so on and so forth. So there are fewer receptors, but they recognize common elements. That's the point that I'm trying to drive home, so that there isn't huge diversity in the number of number of receptors that are there, but they recognize something that's common in the pathogens. So there is discrimination between pathogens of a kind, and here something is gone off. Anyway, so this is the macrophage which is sort of superimposed, and what you see is TLR2 and TLR4, for example. So all macrophages which will have TLR4, and all TLR4s will have identical structure, and all TLR4s will recognize more or less identical structure. For example, a TLR4 is a receptor for lipopolysaccharides. So E. coli as gram-negatives have lipopolysaccharides, Salmonella typhi, other species of Salmonella, all of these have lipopolysaccharides. All of those will be recognized by TLR4. Right? So that's what I meant, that many diverse pathogens, but common receptors. The need of the hour is that the receptor should be present on the cell, immediately latch on to the pathogen. That's the innate immune function, and that's why we have innate immune system receptors of this kind. And this is a very different situation as compared to what I'm going to show you now for the rest of my talk as adaptive immune cell recognition. This is a very complex process, and I had difficulty when I teach. As I said, I primarily teach these days, but I use at least 3 full hours, not even 40 minutes, 3 full hours to teach this, if not more. So it's compressing, trying to give you a sense, but I hope I succeed to an extent. So T and B cells are the most critical components of the adaptive system, which use the kind of receptors that I'm going to talk about. And they are capable, B cell as a subset in the body, T cell as another subset in the body, they are capable of recognizing 10 million plus individual specificities in humans at any given time point. So I am not exaggerating, 10 million is the diversity, which means 10 million different antigens, what we would call in a better immunological term as epitopes. So 10 million different epitopes can be recognized by B cells with the help of their receptors, and T cells with the help of their receptors. That's the diversity. That's the diversity of pathogen recognition that I'm talking about, unlike the innate immune cells where I mentioned a few receptors which are common, which share the common structure. So there is quite a bit of difference between the recognition system. So recognition of proteins, polysaccharides, peptides, all of this can be done between the B cell and T cells, and they are from individual pathogens, unlike innate immune cells, which is what I just mentioned. So there is also daily production of new B cells and new T cells, lot fewer in me because I'm old, much more in you because you are young, because organs like thymus, I'll show you eventually that they start shrinking in terms of their production capabilities, but essentially new T cells and B cells are produced every day. So this 10 million that I'm talking about, if I'm exposed to a pathogen and 50 different T cells got used up, as going towards effector immune response, there will be newer ones generated. So at any given time point, especially in young individuals, this is not my phone, the young individuals who will have this diversity in them. So this is something that is useful as to know in the context of immunological memory, about which I'm not going to talk, but I am referring to it because that's the width of the immunological memory that we are endowed with potentially. So I'm going to give you a sense of B cell receptor and T cell receptor. These are two separate entities. There are commonalities in the structure, there are differences and the function. So this is something that's sort of, introductory for you to remember. So in the B cells, there are two identical B cell receptor, two identical mu heavy chains, which combine with two identical either kappa or lambda light chains to form a receptor. So it's a two-chain based receptor, but as you know, antibodies are normally drawn as Y with the two arms, which we call, that's the two identical chains that I'm talking about. In contrast, T cell receptor has only one beta chain and one alpha chain, which combines to form a receptor. Heavy chain combines first, followed, and it has V, D, and J chains. And this V, D, J chains is important, and I'm going to show you the cartoon for that. In here, beta chain combines first and again has V, D, J, and the C is the constant chain. After the VDJ, so heavy chain recombination is over, there is a kappa light chain which combines next, and it is a light chain, so it does not have all three mini genes that I'll show you in a minute. It has only VJ. Similarly, alpha chain has only VJ. And the kappa light chain, for whatever reason, if it fails to combine, then the lambda light chain complexes also, lambda light chain locus also gets activated, and it will fill up the position of kappa in case kappa fills. So this is, as I said, sort of broad comparison. This is how the B cell receptor looks. And normally you may think of B cell or the specificity that we talk about as antibodies, but primarily B cell receptor is a membrane anchored receptor, and when the B cell gets activated, the effector form of it is the antibody, which is secreted. So right now we are talking about the cell surface anchored receptor. This is how it looks like, and the red circles that you see are the areas where the receptor specificity lies, and here this particular structure that we are talking about, they are identical. So this is the heavy chain and this is the light chain. So this is also the heavy chain and the light chain. So two heavy chains are identical, two light chains are identical. So the receptors are identical. But what I said was, on a given B cell, this is a B cell receptor. And we have 10 million different B cells which can recognize different antigens. So this is the T cell receptor structure again. And here you will notice that alpha chain, beta chain together form a B cell receptor. And again, that also is membrane anchored. So this is how the receptors look before they are getting activated. And as I said, you people might be familiar with this B cell receptor getting ultimately converted into the antibody secreting molecule. This is simply to show you that how different B cell receptors would recognize different elements, and you can imagine that it could be a very small peptide that we were talking about, Arvind was talking about. Even that small peptide can be recognized by B cells. It could be different shapes of the antigen that are very much there. The last one that is shown here is that B cells can also recognize part of the protein. So protein will have its own contours, and some part of it will be latched on by one B cell, another part might be latched on by the other B cell. So one single protein antigen that we are talking about may have potentially 5, 6, 7, 10, depending on the size and the shape, different B cell binding capacity. So one protein here is shown to have this particular B cell receptor binding to it, but another B cell might be capable of latching at this end or that end. That's how, for a single protein, we have variety of B cell receptor mediated interactions that we think of. So this is for T cells. So what I showed you previously here is that B cell is directly latching onto the antigen and recognizing. It is not recognizing the whole protein necessarily, but it can recognize small portions of it, which is what we call as epitopes. So here there is the soluble portion. So B cells can recognize soluble proteins or cell surface associated proteins or polysaccharides and so on and so forth. For the T cell, the recognition itself is different. It requires a two-component system. And the first component of that is this MHC molecule, major histocompatibility complex molecule. And in humans, the system is called HLA. HLA, so class 1 HLA in humans is ABC. So this is the heavy chain, this is the light chain. And what you see here fitting is the peptide in the peptide binding groove. The other half is the MHC class 2. In humans, it is HLA, DR, DP, DQ, and so on and so forth. Let's not go into the details of that because that's not relevant here. But what is shown now is that this component, so MHC, whether it's class 1 or class 2, is my own self molecules. And what T cells are supposed to recognize is this foreign peptide, so-called foreign peptide, which is a non-self, which is fitting into the class 2 or class 1, different peptides possibly, but it's a self plus non-self together, which is being recognized by T cells. Arvind was making a reference to self-non-self recognition, and he started with the first slide where complement doesn't actually do non-self recognition distinction, but there are regulators. Here, what you see is that this is the self component, this is the foreign component, and what T cell recognizes is this portion. So there will be part of the MHC, part of the so-called upper portion of the peptide, if it is, say, in the helical form, as well as the other chain. So that's what will be recognized by the T cell receptor. This is the end-on view that I haven't put in, but that's what we need to think of. So how is this diversity generated? We have looked at the ligands that B cells recognize. We have looked at the ligands that T cells recognize. And I know there are going to be following classes where MHC, antigen processing, presentation, and so on will be taught to you or talked to you. So that portion I'm going to leave out. But how is the diversity generated within the constraints of the genome? Because you can imagine if 10 million different combinations have to be generated, those many segments or potential genes should be there in the T cell, on our chromosome, is what you would think of, which is not how it works, because what we have is So each half of the two-component receptor is generated from mini genes. So it is not one full VDJ which is available and put out onto the cell surface, but we have V, D, and J, which are separate sets of mini gene along with constant gene. And there are so-called permutations and combinations to give rise potentially a diversity of 10 to the power 12. So when I was talking about 10 to the power 7, 10 million different diversities which are available at any time point, this is the potential that our genome can generate, of which every time 10 to 12 is not generated. Whatever is generated is random, and I'll show you in a minute how that randomness is very much there. So take this as an example. I hope you can see different colors. This is so-called V, or the variable portion, D, or the diversity portion, and J along with the C. And what is shown here is the permutation combination that I was talking about. So first, D and J join in the heavy chain in any context. So out of these, any one will be chosen. Out of these, any one will be chosen. So we have one DJ combination. Assume it is D1 and J1. Okay, and now we have so many V genes, and I'm going to show you approximate numbers in the next couple of slides. But V1, J1 can combine with V1, V2, V3, V4, V5, V7, V8, and so on and so forth. So that's how the diversity can be generated with the sets of genes. If it is J1 which combines with D2, we are thinking of a different receptor emerging out. We are thinking of J1 and D2 with V7, for example, another receptor. So that's how we are talking about how the diversity is generated. And this diversity, or recombination, I should say, recombination is generated, and the enzymes responsible for that are called recombinase activating gene, or RAG1 and RAG2. That's the very, very critical part. If there are unfortunate individuals who have deficiency in RAG1, RAG2 mutations, and they are really, really extremely sensitive, most of them die soon after birth because there are no B cells and no T cells which come out of their bone marrow and the thymus. Look at this available number. If there is any Shakuntala Devi equivalent, you can do all the permutation combination and tell me whether it actually matches to 10 raised to the power 12. I haven't attempted it. I must admit that I've forgotten the formulas for the calculation also. But for B cells, in the heavy chain region, variable genes are 65, and between kappa and lambda, there will be, which is light chain, there will be 70 V genes. D is only present in heavy chain locus, so there will be 27. In the J, heavy chain, 6, kappa lambda 9. So from here, you have to calculate the permutations combinations. For T cells, again, you see the numbers. Numbers are huge. So this is the mini gene. So we only have inheritance of the mini genes. And then the combination will happen, which is in a sense random. And that's why when T cell receptors and B cell receptors are generated, they are randomly generated. At that time, there is no sense, I mean, immune system is, we can't think of it as human being which can think and move on. It is a random process. It's a molecular process of recombination, and we have diversity of receptors which will be available to us. This is a very old report, but mind you, by now these kinds of data are not so outdated because the gene sequencing and everything is available. So I put the data here without worrying about it. This is going through the details of the process, and now you will realize how complicated it is and how challenging the job is for me. Well, more than me, for you to follow, which means the challenge is thrown at me to explain things better. But if this is simply an example that if we are, this is, forget about the details of this, but this is the earliest cell that we are talking about. B cells develop in the bone marrow, T cells in the thymus, I'm sure you know that. But what happens is that first, heavy chain gene rearrangement starts. We have two chromosomes with identical sets, not identical, but sets of genes which are more or less the same VDJs. There will be similar VDJs will be very much there. And the first recombination has to be D and J starting on both chromosomes. If one runs ahead a little bit and is successful, there is a negative feedback. So we don't have two DJs coming out of DJ is not what we heard last night. This is the D and J regions coming together, which will be very much there. And there will be only one DJ recombined in a given cell. If it is successful, on the first chromosome, things move on. If this is a failure, which is what this negative arrow shows, then the second chromosome recombination will continue, and if that also is not successful, there will be cell loss. So there are options of generating more than one. So similarly, then the light chain gene, and you can imagine that first kappa gene on first chromosome, failure, second chromosome, failure, then lambda gene on the first chromosome, lambda gene on the second chromosome, and cell loss. So there is so much of variety of recombination available. As a result, finally, you will have either the kappa chain expressing mu heavy chain in recombined B cell receptor, IgM, or the lambda chain combining IgM. And I must tell you one more component of the diversity which is mentioned here. When there is this heavy chain recombination which is complete, so there is one heavy chain, there isn't a light chain which has started recombining, but this B cell at this stage actually undergoes 30 to 60-fold 60-fold increase in numbers, the cell division. As a result, one heavy chain recombined, let's call it an X, that heavy chain recombination has resulted into progeny which has gone through 60 divisions, 50 divisions. And to that, new lambda chain or recombined lambda chain or kappa chain would come up. So that also adds to the diversity, and that's what I meant, that it is enormous generation of diversity that we are talking about. This is about T cells now. We are moving to the T cells, and that's even more complicated, so bear with me. But this is a section of the thymus, and what you will see here is that so-called double negative cells to double positive cells, and then the single positive. That's what is written up here, and there are various cell types which are responsible. Hopefully, I'm going to show you a 3-5 minute movie which will give you more details of that, so bear with me. But there are cortical epithelial cells which have been highlighted, and there are medullary epithelial cells which are also highlighted. So these are epithelial cells specific to the thymus, but they are very critical, and in what we call, what Arvind referred to and I'm going to harp on, is so-called non-self recognition, where the receptors are culled if they are self-reactive. So any self-reacting B cell or any self-reacting T cell is essentially not permitted to get out of the bone marrow or the thymus. And that is why what we get out is only potentially cells which are capable of not recognizing self, but only capable of recognizing non-self. That's the process. I'll show you in, some details of that. But these are the cell types, and I will, when you see the movie, you will realize it, or I'll explain it later. So this I'm going to show you one slide simply to give you a sense that these are all the V betas. There is a D and J beta. So if there is a recombination, great. If not, second one. And finally, after successful or productive rearrangement, there will be production of mRNA and finally protein. The next one I'm going to skip because it's essentially similar. But how does, now this is the serious part, how does randomly generated diversity help only non-self recognition and not self-recognition by the T and B cells? So that is where the critical parts come in. This is about the B cells. We are talking about bone marrow, and what you see here now is this particular B cell which has recombined, and it has IgM receptors on its surface. And it could have one or the other option, or one or the other fate, as one can call it, and I'm only going to describe 2 fates rather than 4. So if this IgM that one sees has no reaction, it does not bind to any protein that is available either on the cell surface or in solution in the bone marrow, which means essentially a non-reactive receptor, if I can call that. So that non-reactive receptor is potentially capable of reacting to and binding to something that is non-self. It's not binding to self, it is potentially. Again, we don't know what non-self it will be. Non-self 1, 2, 3, 4, 10 million. But that is what it will be. And this particular cell will move out to the periphery, and these are the cells which will finally latch on to the targets and start responding to the pathogen presence. The second one that has been shown here is that, say it's a multivalent self molecule, and in that case, this particular antibody is binding to the cell surface proteins, and that is very high intensity reaction. As a result, it is a very clearly self-reactive B cell, and hence it is dangerous. As a result, this cell poses a risk, so this cell should actually be killed off. Or there is another process, and it simply says, and I will mention it, not go into the details of that, but either there will be deletion of this clone, this B cell, or there will be further modification possible so that this receptor changes from whatever it is to whatever the new one. So lambda light chain or kappa light chain would undergo another round of recombination to modify the receptor. And if this receptor is good, not self-reactive, it will move on. If it is still self-reactive, it will be culled off. That's how the B cell process goes through. This is the film that I hope you will be able to.
T cell development takes place within the thymus, a specialized organ where T cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations, and as thymocytes mature, they pass from one environment to another. T cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start their process of maturation by rearrangement of their T cell receptor genes. As maturation proceeds and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocytes express both CD4 and CD8, as well as an alpha-beta T cell receptor. A cell that has a receptor able to recognize an MHC class I molecule receives both a survival signal and a maturation signal. Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class II molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class I or MHC class II molecules fail to receive any survival signals and die by programmed cell death or apoptosis. Thymocytes that are able to recognize MHC class I or MHC class II peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system. T cell development takes place within the thymus, a specialized organ where
Sorry, I thought I probably pressed wrong one. Okay, so that was it. I know this is very textbook, but it gives you a sense of how much selection process is undergoing in the thymus. And this is a recap of the positive selection, that you do need to have interaction with so no binding, cell will die. You do need to have interaction with your own self-MHC molecules with certain avidity, then only the cells would move on as positive selection. And if you are looking at the next one, that the moderate binding lives. If there is very high binding, it dies, because this is a self-reactive T cell. That's the point. There are nuances in that picture which you may or may not be able to see, but this is what I wanted to show, that where I fall, and many of the teachers that you have, not everybody, who we fall up there. So we have 10% production of what we had, all of us had at our birth. So the thymic output daily goes down, but I'm surviving, many of us are surviving, our lifespan is increasing. So obviously, even if there is this low level of fresh, naive cell production, we are capable of living. What you must remember, though, is that during COVID, elderly people got preference for vaccination. That is because as you become old, you definitely become more and more susceptible to infections and sicknesses which follow. And now you know the reason that our output is so low. So that's why the diversity shrinks, and as a result, we become more and more susceptible. You will become one day too. So there is one last part which I'm simply going to quickly go through, and then give you some other interesting things about as take homes. Cell with negative feedback potential. These are the regulatory T cells which are also controlling the system that we have. We don't want very high inflammation, and at that point such regulatory cells will come into picture. I will really not go through the details because I think I'm running out of time, but now let's look at, this is my attempt to figure out why you need to know the immune diversity. What are the advantages of the immune diversity? So innate cells, as you know, one receptor and many targets, and we do need rapid response, so that limited diversity, but diversity is also very important. And exquisite pathogen-specific response, that also I mentioned just now. How they respond, I did not, but the diversity bit, I hope, is clear. It is rare indeed to find individuals with inability to recognize a given pathogen. And it covers the known and the unknown universe of pathogen. For human beings, SARS-CoV-2 was the unknown until whatever, December 19 or so, and all of us more or less mounted very good immune response. That is just one example how any shape of a pathogen can eventually be recognized by B cells and T cells at different time points. So innate and adaptive combination is a continuum of immune response development from minutes to days, and there is an advantage to populate at a population level. Pathogens tackled well by subgroups. There is no extinction due to pathogen dominance, and this may get covered when you hear about the antigen processing presentation and HLA diversity. And this is a question which bothered me as the last slide. What can be the utility of talking to you about the diversity of immune repertoire? So, few bits. Boosting of immunological memory, and you know it has to be antigen-specific approaches rather than general approaches. Otherwise, there is a wastage. So I have, during COVID again, there were these general immune boosters, completely useless. In fact, they will cause problems because I have, say, typhoid-specific, Japanese encephalitis-specific, malaria-specific memory cells in me. And now you are giving generalized boosters. What was needed was SARS-CoV-2 specific response. Why should other memory T cells get activated and churn out cytokines? Useless. In fact, if you trigger them unnecessarily, this is unnecessary trigger, then you are decreasing the utility of immunological memory, which should be used appropriately. I suspect I went hoarse telling people not to take these idiotic generalized immunostimulants, but this is the reason. Now you know that there is diversity and you need to trigger only specific antigen-specific response. That's what vaccines do. So you do need to take vaccines, but don't take generalized immunostimulants. So boosting Treg responses as anti-inflammatory and immunosuppressive approach, this is something that is useful, and there are approaches which are getting developed, but those Tregs again develop in the thymus. So in the cancer context, identifying cancer-specific T cells and then expanding them in vitro, this is in the future. This is not so common. It's a bit of it has started coming up. These are not CAR T cells. Okay, we are not talking about current CAR T cell therapy, which became available at a very high cost in India also, was directed against all B cells. So, but that was a cancer situation. So this is very different from what I mentioned in the first one, which is the antigen specificity. There is also a possibility of modifying Th1, Th2 balance in antigen-specific manner. This is something that maybe Amit will talk or maybe not talk, but there is therapeutic approaches for allergy and autoimmune diseases. So this is potential applications of what I said remotely. Thank you very much. That was the last one, right?
Okay, many thanks, Vineeta, for that whirlwind coverage of so many things. Questions? Okay, let me start out with one. So, you said that these antibody VDJ combination is random. So, but when, certainly with viral pathogens, it's not that doesn't seem like you get a huge diversity of germline VH genes which actually come up finally. It's very often it's a similar, like a VH1/2 for different pathogens.
Yes.
So why is that?
So that is very difficult to answer in the sense that in terms of recombination, sometimes one can say that why certain VHs are preferred. If you're looking at the chromosomal location, then some VHs become preferred because they are closer to the recombination site and so on. So we do see some heavy presence of certain VHs as compared to certain others. So what I was talking about was really the randomness, that if all were available and all were equally chosen, that's the capacity that we have. Your question was specifically in the context of some VH genes. I don't think anybody has an explanation, because for one virus it could be VH17 versus something else. On the other, for some other viruses, there is also another heavy chain subset which gets expanded. The point is, I don't know, and this is something where one needs to look at, that the individuals who are responding with one particular subset, how is their repertoire looking? Okay, in their repertoire, if that particular heavy gene family is overrepresented, then for any immune response, you will get that. But in another individual, if say, instead of heavy chain V5, in another individual, if it is heavy chain V17, then are they still responding to the same pathogen with fifth family as the dominant? These are the nuanced questions that we need to ask, and I don't think, well, I should not say it hasn't been done. I don't think I have actually looked for data, looked for papers which explain this. But that is the logical explanation that I'm talking about.
Thanks, ma'am, for the wonderful talk. For T cells, we kind of know that for any given epitope, so you could get, let's say, between 100 to 500 specificities. So
antigen, epitope is,
yeah.
Epitope is difficult.
So what is this number for B cells?
Did Dinkar talk about it yesterday? No, but it is quite similar. But because again, for T cells, when we talk about the epitopes, we are talking about different peptides which get loaded and hence different shapes, or the same peptide on different MHCs and hence different shape, right? Essentially, MHC peptide complex forms a three-dimensional interacting surface, and to which the T cell receptor latches on. So that surface is what, as you said, for one protein, it could be anywhere 100 to 500. B cells, depending on the structure of the antigen, because that will be much more variable than the T cell. It could be polysaccharide, it could be a protein antigen, it could be the whole bacterium and parts of that. And again, what you have to remember is that it will not be a linear epitope, it could be the conformational epitope as well. So with that, I suspect that, and looking at what IgM binds with low avidity, probably the number would be higher than the T cell. That's my guess. But eventually, the avidity will mature, and finally, when from IgM, I'm sorry, this is immunology, IgM to IgG, when it moves over, there will be fewer epitopes. I had to bring in something additional which I haven't talked about.
Greetings of the day, ma'am. It was a wonderful talk. So my question is that I want to know about we talked about 10 million different possibilities of the T cells and the B cell receptors. Now, as we age, the thymus is depleting, and of course, in our body, there is presence of the diverse T cells and B cells which have not been exposed to antigens.
Perfect.
So, how is it that, is there any selection or choice of some antigens, like the cells, the memory cells which have not been exposed to antigens, what is the diversity at the later ages?
Okay, let me paraphrase the question because you mixed up two issues. What I was talking about is the naive T cell repertoire as it comes out of the thymus. Same logic for B cell, let's restrict to T cells since you asked. So naive T cell repertoire is of 10 million. If there is massive viral infection, there will be, say, 1,000 T cells out of this 10 million, which were directed against the virus, so they stopped being naive, they became effector or memory cell. That's the depletion of the pool. Over those 4 or 5 days of viral infection, there is still input from the thymus, right? So essentially, the 10 million pool remains 10 million, and the repertoire or the diversity which one sees in the effector and memory cells is excluded from this in the first place. Okay, that diversity is required, and that's why I talked about immunological memory for the significance of what I'm saying. But that is not what is included. So with advancing age, even from 10 million, it may go down theoretically to 1 million. 1 million is also large. So if against a pathogen, instead of 100 and 150 different T cells coming up and moving from the naive pool to the memory pool or effector pool, there will still be cells which will be very much available in the naive repertoire, which will start responding. So that's one way. What happens actually is because the size of the naive cell pool decreases, some cells from the memory pool automatically expand. And this has been shown in the CD8 context for one of the viruses very commonly in much more in developed world because the data are available in the developed world and not here, but memory cell pool unnecessarily increases. Okay, because there is space. So we are talking about niche in the lymph node, area in the lymph node where T cells should be. If the naive pool is shrinking, maybe existing memory cells would simply expand in terms of number and occupy. So there is clear imbalance in the pool. One should also remember that if an naive cell is produced today and it wanders around the whole body searching in terms of its targets, and say in another 100 days or 200 days, there is no target, this cell is not an immortal cell. In a sense, it becomes old, senescent, and it's killed off. So the naive cell pool is not so old in the young ones because there is good production. Naive cell pool can become somewhat old in somebody like me because there isn't enough naive cell push from the thymus, no output, not so much output from the thymus, that the existing pool is pushed off. Okay, so there is a balance and there is an imbalance, but still we survive.
MSc discriminates between self and non-self. So, I try to differ, with that definition. I just want Madam to give a very simplistic explanation to this statement. Or maybe,
I would say that actual discrimination begins for T cells, since you are talking about MHC, at the time of thymic selection process that I showed you. So, for example, all of you know that there is a transplant rejection, kidney transplant and whatever, and most people are on immunosuppressives. The reason is that the foreign MHC has been introduced into your body. The kidney cells are not just kidney cells. Every kidney cell would have MHC molecules on its surface. So it's a foreign MHC which has been, which is seen as foreign. All right, so which means that my T cells, as they came out of the thymus, were capable of recognizing my MHC molecules as mine, and what was not mine will be foreign. So that is why there is a need, and ideally, which idealism is, it fails, transplant should be as MHC matched as possible. So if there is MHC, HLA-DR2 here in me, if there is HLA-DR2 in the graft, then that MHC is treated as mine. sort of, and there is less response mounted against that. But if there are, say, DR7 and I don't have DR7, that is treated as foreign. So it is not MHC which tells us to distinguish between me and you, but it is T cells which respond to presence of foreign MHC so that there is a transplant rejection. And there are historically wonderful experiments when we didn't know even about MHC, but that is history of immunology, which is an interesting and separate topic itself. But people there is a horrible, horrible, horrible picture of a black man with a white leg transplanted, okay? So, I mean, it's not just because it's black and white that it feels contrasting, but any man's leg was put onto there, and I'm sure it was rejected in no time. Because at that time, there was only the skill was plastic surgery. We had plastic surgery skill even in Ganpati's days, but this is reality, okay? We claim that we had plastic surgery skill to get that elephant head onto a god, but that's a separate one. Even there, if it was an elephant head which was transplanted, having plastic surgery, the head would be rejected because there was MHC incompatibility. We don't think of that.
So I just need one more
Sorry,
clarification.
I hope that was sufficient.
It is related to what Sarath asked. You have a transplanted tissue, and you said that kidney cells, they express a foreign HLA, right? So our MHC will recognize that and reject. But then there is a concept of MHC restriction where there has to be self and non-self antigen has to be presented by a self MHC, and the antigen presenting cell or the T cell receptor has to see both together. So here it is not presented like that, isn't it?
Agreed. Shall we go into deeper immunology? I think people will get bored. This is what is called also as allorecognition, so that this is a foreign MHC which is recognized. So what I said was that there is MHC with a fitted peptide. That shape is being recognized, right? So MHC identification was necessary for the positive selection in the thymus. That is what he's calling as MHC restriction. So that's part of our T cell repertoire. It is accepted. That's not something that worries us. But now if the MHC peptide complex is having a different MHC, but say same albumin-derived peptide, it could be the same identical peptide, it could be a different peptide, but that in itself is something that is a problem. So that is why we recognize foreign grafts in two ways. The foreign proteins, I mean, you have an albumin and I have albumin, but what albumin peptides I generate because of various processing which he is going to talk about versus anybody else generates could be different. So even self-peptides which are loaded onto the MHC could be different. I'm sorry, I'm going into Greek and Latin. Probably I'll stop and answer him outside. So, but what is being recognized is called allorecognition or foreign MHC recognition, and that is why the transplant tissues get rejected. And there are many nuances of transplant rejection, which I will not go into the details because I think I've already given an overdose.
Okay, just one more out of the many things I didn't understand. When this rearrangement is happening, how does one chromosome know what is happening on the other one?
For that, there are actually molecular feedbacks. So as I said, when
it's a stochastic process.
Initial process is stochastic, but when which VDJ is getting chosen by one chromosome, if it is more efficiently done, then the recombination would happen, which provides the signal for the other chromosomal recombination to stop. So processes more or less begin really in parallel together. But if there is so much of V, depends on which V is selected here, it might take a little extra time. I know we are talking about small differences in time, but still that matters.
Okay then, thank you very much, Vineeta.
Thank you, ma'am, for the interesting talk. And I request Raghavan sir to present the memento to Dr. Vineeta.
Thank you so much.
I would also like to thank Dr. Raghavan for chairing the session. And I request Dr. Bhagwati Saxena to felicitate Dr. Raghavan. We kindly request everyone to proceed for the tea break, and we will resume back at 12:10 p.m.